This time we will start a new topic, that's plant tissue culture and technique. What is plant tissue culture? Plant tissue culture is defined as culturing plant seed, organ, explant, tissue, cell, or protoplasm on a chemical defined synthetic nutrient media under sterile and controlled condition of light, temperature, and humidity. Plant tissue culture is used to describe the in vitro and aseptic growth of any plant part on a tissue culture medium. This technology is based on three fundamental principles. The first one, the plant part or explant must be selected and isolated from the rest of plant body. The second, the explant must be maintained in control physically and chemically defined condition. And explant, the tissue taken from a selected plant are transferred to a culture medium open to establish a new plant. There are several advantages of plant tissue culture, such as production of exact copies of plant that produce particularly a good flower, a fruit, and have other desirable traits. To quickly mature a plant, production of multiple of plant in absence of seed or necessary pollinator to produce a seed, the regeneration of whole plant cells that have been genetically modified, the production of plant in sterile conditioner, in sterile container that allow them to move with greatly reduced chance of transmitting disease, pest, and pathogen, and to clean particular plant of viral and other infections, and to quickly multiply this plant as clean stock for horticulture and agriculture. Now let's see the history of plant tissue culture. The first one in 1902, the idea of totipotency of plant cell. This is introduced by Herbert Allan. In 1937, White first time established successful root culture of tomato. Then in 1941, Van Verbeek used coconut meat for growth and development of young Dantur embryo. In 1957, Ascott and Miller demonstrated the role of auxin and cytokine on root and stalk formation in tobacco tissue. 1962, Morisage and Scott introduced the medium for tobacco culture, and in 1987, isolation of Bt gene from Bacterium Bacillus thuringiensis. Now let's move to principle of plant tissue culture. Several principles in plant tissue culture. Plant tissue culture rely on the fact that many plant cells have the ability to regenerate a whole plant, or we call that one as totipotency. Single cell, plant cell without a cell wall, we call this one as a protoplasm. Piece of leaf, stems, or root can often be used to generate a new plant on culture media given required nutrient and plant hormone. And the last, the control condition provide the culture and environmental conducive for growth and multiplication. Plant tissue culture are generally initiated from multicellular tissue fragment called explant. Explant are obtained from a living plant. Explant may originate from a wide range of plant tissue, such as leaf, stem, root, petiole, hypocotyl, cotyledon, embryo, or meristem. In biology, explant culture is a technique to organotypically culture cell from a piece of tissue or organ removed from a plant or animal. The term explant can be applied to a sample obtained from any part of the organism. The plant growth can be achieved in two ways: sought directly by appropriate media and by somatic embryogenesis. There are several techniques involved in plant tissue culture, such as sterilization, media preparation, culture condition, induction of callus, embryogenesis, and hardening. Let's see this figure in the right side from this one. Then we'll get a new plant to regenerate this carrot. Now let's move to sterilization. Sterilization is
The first is sterilization. Sterilization is the technique employed to get rid of microbes, such as bacteria and fungi, in the culture medium, vessels, and explant. There are several steps in the sterilization in plant tissue culture. The first is maintenance of aseptic environment. During the in vitro tissue culture, maintenance of aseptic environmental conditions should be followed, such as sterilization of glassware, forceps, scalpels, and all accessories in wet steam sterilization. The second, sterilization of culture room. Floor and walls are washed first with detergent and then with 2% sodium hypochlorite or 95% ethanol. The cabinet of laminar airflow is sterilized by clearing the work surface with 95% ethanol and then exposure of UV radiation for 15 minutes. Sterilization of nutrient media. Culture media are dispensed in glass containers, plugged with non-absorbent cotton or sealed with plastic closure, and then sterilized using autoclave at 15 psi or 121 degrees Celsius for 15 to 30 minutes. The last, sterilization of explant. The plant material to be used for tissue culture should be surface sterilized by first exposing the material in running tap water and then treating it in surface sterilization agent like 0.1% mercury chloride, 70% ethanol under aseptic condition inside the laminar airflow chamber. Sterilization is achieved by one of the following approaches, such as dry heat treatment, flame sterilization, autoclaving, filter sterilization, wiping with 70% ethanol, and surface sterilization. We move to the media preparation. Media preparation. Plant tissue culture media are generally composed of inorganic element, organic compound, and support matrix. Culture media provide the culture the necessary inorganic nutrients that are usually available from soil. In addition, they also provide the culture the necessary organic compounds such as vitamins, carbon source, which are usually produced in plants. Sometimes, plant growth regulator are added to the medium to stimulate cell diffusion and or differentiation. There are several major types of culture media such as white media. White media is one of the earliest plant tissue culture media. It has a lower salt concentration and higher concentration of magnesium sulfate. The concentration of nitrate is 19% lesser than the MS media. White medium can be used for the purpose of shot culture and callus culture. It is suitable for culture Musa and Daucus species. The second is MS medium. MS medium formulated by Muragise and Scoop is most widely used for many types of culture system. This medium is used to induce organogenesis, callus culture, micropropagation, and cell suspension. The next is B5 media. B5 media developed by Gambor for cell suspension and callus, and recently it is modified for use for protoplast culture. The medium has higher concentration of nitrate and potassium and a lower concentration of ammonia. Potassium nitrate is useful in inducing the soybean root callus formation, and ammonium sulfate plays an essential role in cell growth. It is used for the purpose of protoplast culture. Linsmeyer and Cook or LS media. This medium was developed by Linsmeyer and Cook in 1965. It was first used to optimize the organic supplement of the tobacco culture. The medium has a similar component as Murasige and Cook, with a twist of Linsmeyer and Cook vitamin. It is used for the purpose of organogenesis, callus culture, cell suspension, and micropropagation. The last is niche media. The medium was developed by G.P. Niche in 1969 for establishment of the in vitro culture of Nicotiana from family Solanaceae. It contained a high concentration of thiamine, biotin, and folic acid that support anthercallus. This medium is used to establish the in vitro anthercallus. Next, the culture condition. Commonly, there are several factors which affect two culture conditions, such as pH, normally adjusted between 5.6 to 6. The second is temperature, incubate normally at constant temperature of 250 degrees Celsius for optimal growth. The third is humidity and light intensity. The culture requires 50 up to 60% relative humidity and 16 hours of photoperiod by illumination of cool white fluorescence tubes of approximately 1000 looks. The last, aeration. Aeration to the culture can be provided by shaking the flask or tubes of liquid culture on automatic shaker, or aeration of the medium by passing with filter sterilized air. The next factor is induction of callus. Plant callus, or plural callus or kali, is growing mass of unorganized plant parenchyma cell. Explant of 1 up to 2 cm sterile segments selected from leaf, stem, tuber, or root inoculate in the MS nutrient medium, supplemented with auxin and incubated at 25 degrees Celsius and supplement in an alternate light and dark period of 12 hours to induce cell division, and soon the upper surface of explant develop into callus. The next factor is embryogenesis. The process of formation of an embryo is called embryogenesis. Embryogenesis starts from a single embryogenic cell that can be a zygote, that product of the fusion of an egg and a sperm during fertilization, or in undifferentiated callus cell. Embryos developing from zygote are called zygotic embryos, while those derived from somatic cell are called somatic embryos. During the embryonic development, the polar axis of the plant is established. Domain that set up the organization of the plant body are defined, and the primary tissue and organ system are delineated. Somatic embryogenesis is another important way to regenerate new plant in plant tissue culture. Embryo development is bipolar, or having a shoot pole and a radicular pole at opposite end. Embryos are not organs because they are structurally independent from their parent body, such as they do not have a vascular system connecting them with their parent plant body. The last factor that involved in technique of plant tissue culture is hardening. Hardening is the incubation of plant is neat plastic pouch for 6 up to 8 weeks in greenhouse. These plants are incubated in a liquid nutrient medium. Therefore, hardening is the acclimatization of tissue culture plant slowly before growing in the field. Hardening method is a method in which the tissue culture plants developed in artificial media are habituated to grow in natural environment. This picture shows the plant tissue culture procedure. The tissue culture is performed within aseptic condition under the filtered air provided by a laminar flow cabinet. The plant tissue culture technique is accomplished in following steps. The first, initiation state. In this step, the desired tissue is collected, added, and sterilized to hinder the method from any contamination. The second step, multiplication state. In the second step of plant tissue culture, the sterilization explant is included within a tissue culture medium, which is constituted of growth regulators and suitable nutrients. The multiplication of cell occurs within this nutrient-rich medium. This undifferentiated portion of cell is termed as a callus. The next step is a formation of root. The root formation starts different plant growth hormone, including initiation the development of roots. The next step is the formation of shoot. In this step, the plant growth hormone are included, which help in the development of shoot. The shoot formation will be observed for a week. The last step is acclimatization. The plant is transferred into a greenhouse when the plant starts to develop. It will provide controlled environmental conditions for proper development. After that, the plant is sent to a nursery where it grows under natural environmental conditions. Based on the explant, some plant tissue culture types, such as the first organ culture. The culture organ mixed from embryo, anthers, ovary, roots, shoot, or other organ or plant on culture media. The meristem culture from the culture of any plant meristematic tissue on culture media. The third is protoplast culture. Protoplasts are cells without a cell wall but bounded by a cell membrane or plasma membrane. Using protoplasts, it is possible to regenerate whole plants from single cell and also develop somatic hybrid, the step involved in protoplast culture. The last is cell culture. The growing of cell, including the culture of single cell or small aggregate of cell in vitro in liquid medium, is known as cell suspension culture. The cell suspension is prepared by transferring a portion of callus to the liquid medium and agitated using rotary shaker instrument. The cells are separated from the callus tissue and used for cell suspension culture. There are several applications of plant tissue culture. The commercial production of plant used as potting, landscape, and florist subject to conserve rare or endangered plant species, to screen cell rather than plant for advantage character like herbicide resistance and tolerance, large-scale growth of plant cell in liquid culture in bioreactor for production of valuable compounds, to cross-distantly related species by protoplast fusion and regeneration of novel hybrid, to produce clean plant material from stock infected by viruses or other pathogen, and the last to produce the identical sterile hybrid species.